Welcome to this. This is set for their height. Welcome to SIGGRAPH Next. This is a brand new program that we're starting this year. We'll do some experimentation for about 3 years and then roll it into the formal program. And a lot of your feedback helps direct how this goes. So thank you so much for coming. The idea behind SIGGRAPH Next is what's next for SIGGRAPH. So it's very creatively titled. We're in an industry that we are moving forward. You know, how many people are 20, 30-year attendees to the conference, right? We've seen some amazing things, right? When I was in grad school, nobody could render smoke. So we're in many ways responsible. We have the opportunity and both the responsibility of moving the industry forward. So SIGGRAPH Next very specifically focuses on what skill sets that are already core to our community are out in other universes doing really interesting things. So yesterday we had Bob Nichols here from Blizzard, host a representative from the Institute for the Future, talking about moving forward across economies, across emerging technologies, across social norms. Today we have Daniel and Michel are going to talk about medicine. How are we using graphics and how are they being applied into the medical field? Tomorrow we have Tony DeRose, who's with R&D at Pixar, talking about next-gen education. He's done Pixar in a Box in partnership with Khan Academy, and he's also done a complete curriculum for middle schoolers who want to be makers. This was all curated for this year. Next year we're going to do an open call, so if you have an interesting idea, be sure and look to submit that to participate in SIGGRAPH Next. The session is being recorded. It's their voice and their slides. It's not a video camera on them, and then they'll leave people online. And we'll set up online communities for people who are interested in this topic to talk more with them, to talk with other folks who are interested. We'll have a community manager who helps manage that. And then we can carry on this conversation into next year's session or into similar emerging topics. So ideally, this is a conversation. Feel free to move up close and talk to them. We have mics set up in the aisles, or you can just raise your hand. We have exits in the back. Also, obviously, the door you came in if you need to get in or out. And please feel free to get up and down during the session to help yourself to beverages. So thank you very much. Now, does anyone remember Daniel's pink Cadillac from 1999? Yeah. He had a full-size Cadillac. It was gorgeous in the Emerging Technologies venue. And he's partnered with Michel on some of the work he's doing now. So thank you, gentlemen. Take it away.
Thanks. Thanks, MK. Oh, I should probably press this button. That switch us? Hey, that's me. Hi, I'm Daniel Szeket. Thank you, MK. I'm a designer and a tinkerer of sorts, and I've been in this CG industry for a long time. Where are you going?
Make sure it works.
Oh, I see. That's a good idea, I guess. Yeah, I've been coming to SIGGRAPH since 1991. Actually, I missed the amazing Alias Party 91. Okay, Michel.
Yeah, I'm Michel Nederlof. I'm a physicist by training, and I got lost in the world of digital microscopy about 30 years ago, and have been doing that ever since, looking at cells and tissues. So we are going to try to take you away from rendering clouds and
better mic for you.
Smoke for a while, but instead see what is next in line for computer graphics applications. So we're going to talk about rendering cells and tissues and how the intersection of computer graphics and medicine takes place currently in a variety of medical fields.
Okay.
We're working together with the Oregon Health Science University. We have a small company called Quantitative Imaging System, QI, and the Knight Cancer Institute is where we do most of the research and collaborate with a group of Joe Gray on cancer research. We're also collaborating with KDAP, a software company, developer company, that is helping us making a lot of software in a very short amount of time. So this is Daniel. I'm going to introduce Daniel. He's going to introduce me. So.
It's a really bad idea.
Daniel has an illustrious history in computer graphics. Other than his pink Cadillac, he's been around this conference for a long time, and you might know him. He worked for Alias, designing and building very high-end digital graphics software, and he did animation for about 10 years with Disney. And then he moved into actual hardware acceleration and built demos for ATI. Following that, he had quite a while doing education, trying to teach people how to use all the stuff that he's been building. And then he figured out that it's actually really hard to use, so he worked for a company called Maya, which is in Pittsburgh, Pennsylvania. And Maya is focused on human-centric design, so this is all about human-computer interaction and how you go about designing user interfaces that are more efficient and easier to use.
So Michel started this a long time ago, 30 years ago, at the Max Planck Institute as a physicist. And he got into imaging for the just cell imaging, and just got interested in it at that time, and so started looking into that technology, and has been doing that ever since. And he started 7 companies doing all sorts of things, including drug discovery and helping medical imaging in general, and I've totally, see, this is why it was a bad idea. Where was
Well, the thing is that I try to apply digital imaging and microscopy to a variety of different applications. So starting in an academic setting, moving towards research for a lot of people, making these tools available in digital microscopes.
But way back then, you also worked on some Pixar systems, early Pixar systems.
I thought you were moving ahead already.
I wasn't moving ahead. You worked on some early Pixar systems.
Take this thing away from me. Yeah, at Carnegie Mellon, we actually had the Center for Art and Technology, and we got not the first Pixar machine, but I think the second machine or something like that, sent to the Art and Technology Center at Carnegie Mellon, and they wanted us to render cells and tissues with that. So we did that. It was, of course, a volumetric rendering exercise, so the thing was not at all set up to do volumetric rendering.
Right.
But we had a great time. It was fantastic, huge frame buffer, which at the time was the most difficult thing to get. And so thank you, Pixar. This was in 1987, so it's a while ago. Right. My first SIGGRAPH was not 1889, it was 1989. He thinks I'm old. That's the part.
What was that?
They're not aware. That's when Pixar was hardware.
That's
yes,
right.
it was hardware, yes.
Right.
So, if we're going to have interdisciplinary collaboration, it should be okay to say, I don't know. There are a lot of really smart people in very deep fields, and both of us are examples of that. We studied things that were not, they're not medicine. Neither of us are biologists, but we are able to take the knowledge that we have and apply it to help medicine. And so I know very little about medicine, but I know a lot about software, and I know a lot about how people use it.
Yeah, so it's okay to say I don't know. I was a physicist and I got lost in a biology lab for the longest time. And that was before Wikipedia existed, so I couldn't look anything up. I had to go to the library and read about cells for many, many hours. So this was actually very difficult. But routinely we work together with people that are doing computer programming and biology and medical experts and even clinicians that actually treat patients that actually collaborate with the software group. Oh yeah, so I actually did my thesis on fruit flies. So they have really big salivary glands, and the chromosomes are very interesting to look at. So hence.
So you guys might think, okay, so CG's pictures and film created using computers, and obviously there is some of that, but how are we going to apply all of this visual effect stuff to medicine? Yeah,
so actually medicine and the arts have been connected for many hundreds of years, so this is not a new thing actually. So let's go take a little history lesson. Back in the day, people that were doing medicine and studying medicine, looking through anatomy cadavers, would actually have to draw what they saw, and their findings needed to be meticulously detailed. So they had to be artists as well as scientists in order to do their work. So this is not a new thing. Arts and sciences have been connected. We've just been getting better at it by actually inventing new technologies to see more and
So
see at
there's
a
new
higher
technology.
resolution. So the technology, this is Anthony van Leeuwenhoek. He also studied in Delft in Holland. He invented the microscope. So this little gadget here was the first microscope. He put that up to the light, put some pond water in there, and saw little animals, and was amazed by the fact that there were little animals in this pond flying around.
And the picture that you're seeing here is what that actually looks like. So that's a photograph of it, but at the time there were no photographs. So again, back to the only way he was able to communicate the fact that he'd found these little animals was by illustrating it or showing it to somebody.
Yeah, so we're going to fast forward through history because it'll be a really long session otherwise. But the problem at the time was that these things are not visible actually that well through a microscope. They're about as transparent as a glass of water. So one of the first technologies was to actually use conventional absorptive dyes. Here from a hematoxylin, a log tree sap was used to actually color the tissues before you put them under a microscope, so you can actually generate contrast. And we've spent decades on generating new contrast methods just to get these transparent cells to actually light up or be less transparent under a microscope. So this is a hematoxylin stain, and you can see this is now a digital image of what we look at through a microscope. This is the most common routine pathology test. It's called an H&E stain, hematoxylin, and you can see the nuclei. They're the little purple dots, and you can see red blood cells. They're actually stained red very conveniently. And if you look at the red blood cells in a different way, why do we have this slide again, Daniel?
This slide, you might say, okay, we have computer graphics. This is obviously we have microscopes at this point. We have cameras, but there was no way of getting this image at the time. So even though our pencil got better, we're still rendering out something that we envision, something that we understand, so that we can explain it to other people.
Yeah, so in general, huge amounts of information is generated with the digital images coming out of microscope and other fields, and computer graphics has always been involved in either drawing or rendering or in the computer graphics scene that you just saw, actually making three-dimensional reconstructions out of those things. So there's an explosion of information in images. There's also an explosion of information in genetics because some visionary people a few decades ago set out to actually map our entire human genome, which was a very daring endeavor because at the time they planned it, they couldn't actually do it in less than 100 years. And still they got funding for it, and still they set off and actually started the project in the hopes that the technology would catch up with them and they would be able to finish it sooner than planned. And they did. They did it in 10 years instead of 100 years, and they did it under budget. It cost several $1 billion, of course, but right now we're at the point where we can actually sequence a human genome in about 24 hours of compute time and about $1,000. So we can do this very quickly, and with that explode our amount of information. So to summarize the history lesson, there are some key technologies that at each time a new technology is invented, we gain new insights into cells, tissues, genetics, and that spurs not only an enormous amount of research, but also an enormous amount of data to explore. And so this is an exponential growth of information that we're currently challenged with, and that's where we're going to need your help.
And we added the printing press in there because that's really when information started to disseminate to the masses. And so that's when information started to spread exponentially. So with all this information, why haven't we solved cancer yet?
Yeah, that's a good question. So cancer is really not a single disease is the first problem. It's a complicated set of diseases, and so there is no question why we solved cancer because there are many different forms of it. So also, we actually do not understand the specific mechanisms of cancer. We do not understand specific mechanisms of recurrence of cancer. So there is still a lot of work to be done, despite the fact that there are certain drugs that are effective for a certain amount of time. Many of these treatments are not durable in the very long term. And then as we are developing new drugs, 9 out of 10 of those drug trials actually fail. So what are we going to do about that? In order to eliminate cancer, we really should look at cancer as a great number of diseases. So 80 years ago, leukemia and lymphoma were lumped together as a disease of the blood. Then we figured out it's actually 2 completely different diseases, lymphoma and leukemia. And now we have 80 different types of leukemia. So you can imagine that the drug for the disease of the blood wouldn't work at all for 99% of those patients. You have to treat the specific disease. You also have to really understand how the mechanisms work, so you can design drugs that specifically block that disease and that particular pathway.
So really complicated.
Really complicated.
Just to encapsulate.
Personalized design, if you have a particular patient, you should follow the treatment plan and actually know what the response is of a certain drug specifically on that patient, and then design the treatment plan accordingly. So it's very important to keep a pulse and monitor what the treatment is doing, as well as we can help then the drug companies to be more successful in getting their drugs through clinical trials. If we are more specific about it, and more tailored drugs are going to have a higher success rate.
So, okay, I came into this thinking that this was a biology problem. So why is this not a biology problem? It sounded like a biology problem.
So I think it's a computer software problem for a very large extent. And
you're not biased at all.
I'm not biased at all. But there's a couple of good reasons for it. So this is one reason. You can think of solving the functionality of a cell in our bodies as a computer program. So most of you probably are more familiar with computer programming than biology. So if I compare it to debugging a computer program, you can sort of see that we have a parts list for the human cell. We know exactly all these genes that were sequenced. So we know that there are 3 billion base pairs in our DNA molecules, and there's 20,000 genes, and they lead to the construction of 10 million different proteins. So that's a lot of parts. Those parts then play a role as if they are variables in a computer program. So if you look at this picture, oops, there's a button here. Yes. If you look at this picture, the cell nucleus is the thing in the center here, and the cell itself has an orange boundary. And what's essentially happening is that signals are coming to the cell. They get picked up by a receptor on the cytoplasm membrane. Then they get translated through all these different proteins, and you don't need to know the names. There's no quiz at the end, but these are all names of individual proteins. And remember, there's 10 million of those. So, and essentially, it's like a computer program where they're activated, not activated. But instead of bits, Boolean variables 0 and 1, these things are phosphorylated proteins or non-phosphorylated proteins. But you can still program the same way. It's just that you don't know whether it's a magnetic core or a transistor holding the bit.
This program was started being written millions of years ago.
Yeah, hundreds of millions of years ago. So this is, if you, as a programmer, have worked with old code bases, you'll know there's a lot of dead code. If it was 10 years old, 20, 30 years old, you'll find a lot of dead code in your old software, and it's really difficult to get a grasp on how that software works. So this is really old code, so there is probably an enormous amount of dead code in this mechanism. And so you can imagine 100 million years old code that we got secondhand from an ape is really hard to debug. And it also is very hard to debug because we cannot actually see these individual variables. So if you want to look at the MEC protein, you actually need a microscope and a label, and then you can see is it on or off. And then it's a biological sample, so it may be covered in noise and difficult to debug.
And you're saying dead code as if it's no longer functional, but A, we don't know whether it's functional, and B, if you take out one of these variables, something might break. We don't have a debugger. So.
Yeah, we wish we had a debugger, right? Then you can just step through the code and see what's actually happening. Sure. So that's why we need a huge amount of instrumentation to actually see these variables, and we need a huge amount of compute power to actually debug it, and we need a huge amount of visualization to actually be able to see what is happening in this particular program. This is another way of looking at it. We have this giant part list, and the current state of cancer is sort of this. We know what the parts are, but we don't have the assembly manual that says exactly how these parts are putting back together. We know more or less the gas pedal. If you push it, the car goes faster, but we don't know how the gas pedal is exactly connected to parts of the engine.
There might be 3 gas pedals.
There might be 3 gas pedals. 2 may not be used. Right. You may have genetic mutations, for example. There are 3,000 genetic mutations if you have particular cancer. We have no idea which one is important and which ones are noise. Probably 99% of them are actually noise, so which one is the real one? So, and of course, this is a car. Our bodies are considerably more complex than a Volkswagen, so this parts list is way bigger than what you see there, and then you have all these unused screws that you don't know where they should be assembled in as extra parts.
So basically it's a big data problem, right? This is a up-and-coming topic that we're all hearing a lot more about, and that it's the same kind of problem in biology. And so that makes a lot of sense because while all the information was exponentially growing, we basically discovered tea and coffee. Like, that's as good as we got in the last 10,000 years.
And that sort of defines the big data problem. So it's not big data because it's big file sizes, it's big data because you cannot get your arms around it. So it's a relative term.
Now, while our brains haven't really gotten all that much better, our compute cycles have gotten a lot cheaper. In 2011, we were making more transistors than grains of rice and cheaper. So we are able to compute a lot more, and we're getting faster and faster and faster at it. We just aren't making the connection between all that data and the compute cycles yet. And so I'm not sure how we're going to do that, but that's really the crux of it.
If we could connect it and debug it, we would be much better off. And it's not a simple linear process. We're not going to go one step, have a pipeline of processing, and then get the answer of life that comes out on the other side. Instead, it is really a difficult multidimensional problem. We're getting all these features, genetic, proteomic, architectural features, and we have this enormous multidimensional data space, and what we need to do is try to find the manifolds in that data space to distinguish cancer state from regular state, to distinguish a tumor from metastasizing and not metastasizing.
You might think of these nodes as not just data, but or programs or processes, but also disciplines. So different people thinking deeply about different things, collaborating with each other, coming up with insights, and those insights then have to be fed back into the mess so that other people can understand technology, can understand those insights and work with them and start looping them again. And so this kind of puts us back into this feedback loop. We start off with this discovery of technology, or any discovery of technology, say the microscope, and you need somebody to render that technology, somebody to communicate what people have seen. Then you have a new discovery, and that discovery visualizes reality in some way. And so we say, oh, we don't need to visualize it with art anymore because we have actual visualization. We have the actual reality. And then we get to some new technology that doesn't really visualize that way, say like machine vision, where we're training some machine, and it's seeing everything. It doesn't see just the things we would discern. We've got a brain that has been learning how to see for a long time. Computers not so much, and so they see way too much. And then we give it some deep learning. We teach it some things about how we've learned, maybe let it learn itself. At that point, we build some models. We need to visualize those models, and we're back to visualization, and the artist needs to get involved again.
So we ditched the artist 100 years ago, and now we need them back.
Right. And so when we come across new technology, we're not super good at knowing what to do with it. When the TV came out, it was pretty much radio on a screen. And we did that for a long time. And it took a while before we started evolving that. So radio to television, now we're finally getting outside of this like one-way communication to things like VR and AR. So it takes a while for that evolution to happen.
So in the CG world, there's a sort of a mature pipeline. If you compare that to medical applications, X-ray film would be a good example. Years ago, we were taking film X-rays, and doctors would look at the X-ray by putting it on a light box in their office. Okay, this is not very practical, so digital X-ray came along, and this transition has been fully implemented now. If you go to a hospital, there are no film cameras anymore. So everything is digital. Doctor gets the X-ray on the computer screen. But it's like the radio program playing on a TV. He's still looking at the same X-ray picture. There was no magical new application that came along with that. So take it one step further, we could actually do 3D tomography now. So instead of taking one X-ray, you can take a whole stack of them and build a three-dimensional model and then rotate that on a computer screen. So this is an example of a new application that you could actually do with a new medium as opposed to just using it to display the same image. Now, for the case of pathology, this is more interesting because there's really not a lot more we can get about the individual bones in that X-ray. In the case of pathology, it is a much deeper and richer set of information. So let's follow the same example. We were doing glass slide pathology for almost 200 years now with that tree bark stain that was invented in 1818, I think. There's a paper on that. So this is not very new technology. But that has been the standard. And up till now, most of the biopsies that are done are going on a glass slide, and they're not digital. So slowly, hospitals are taking this digital. And the question is, what is now the next application? Because right now, the clinician is still looking at the results of the slide by actually visually comparing it to his experience in that field. So what is the next application, right? The key is that in a pathology slide, we have a lot more information. So we can drill into each individual cell in a tissue and actually query that for 100 different biomarkers and get all these phosphorylation states of all those variables. Well, 100 of them, all would be 10 million, but at least 100 of them on a single cell as data points. And we can pull in genetic information, and we can pull in further proteomic information. And we can do that in 3D. So now the pathologist has this 100-color image with all that data and information available, and what is he going to do with that, and how is he going to interact with that? And so that's sort of this drive to get a pathology workstation of the future so that the pathologist can get better informed decisions with all that information clearly presented to him in a very short amount of time. He can actually guide the treatment into better directions than we can do now.
So this is from the oil and gas industry, and they've started, mind the pun, drilling into some CG technology so that they can benefit from all of the know-how that computer graphics has. And so now they're applying visualization techniques to some of their data so that they can get some new information, they can use that. And so CG is a very mature, robust, deep community. We have developed a whole bunch of techniques and technologies, and a lot of it is being applied to making movies and entertainment. And so it's very possible to take that same technology just as... I keep doing that. I'm going to not stand there. It's very possible to take that technology and as a whole start applying it to other deep knowledge bases. So, yeah. Yeah,
so we are applying this to pathology, and the current state of the art for tools for researchers is
what researchers make. This is homebrew what researchers make. So
homebrew software. So we set off to build advanced tools to do better. So this is the first step, is to essentially get a Photoshop multi-layer, multicolor application that allows the researchers to move around the 100 different fluorescent colors.
Basically, we're talking about, I mean, these images are, say, 100,000 pixels by 100,000 pixels. They're 16 bits in depth, and we now have the ability to superimpose 100 or so layers of those on top of each other. So there's a lot of data in a single image. And researchers have been kind of cobbling tools together to get that to work. And we've been using Photoshop forever. So, what, 92, 91? We figured, okay, let's not reinvent the wheel. Let's give them some tools that we already know how to make. Then we go, how do we revolutionize that? How do we not just give them some, Photoshop? Because they can get Photoshop and figure out how to make that work. Mm-hmm. And so Obviously better visualizations, that's always useful. Machine vision is, an example of technology that we can give them so they can start seeing information in those images as opposed to just pixels. Because right now, all that they've ever seen was a photo turned into a digital pixel, and then they can look at that pixel. And so give the machine some ability to discern what's what, and then also teach the computer some new tricks so that it can actually learn how to see the things that we need it to see or the things that we are seeing.
Yeah, remember it is a cycle, so it is not the linear process. So it is very important to get the right visualization because that is going to determine the next step in that cycle, right? So here's an example of a piece of tissue. It's colored with 30 different fluorescent dyes. So you see actually fluorescent light showing up here in different colors. And each color is representing a particular biomarker. So we're looking at cytokeratin staining, and we're looking at a nuclear staining in blue. You can essentially do a simple transition and turn different colors on and off. And your brain is actually pretty good at picking up where things are. So if you go back and forth a little bit, you can start to get an idea of the distribution of the biomarkers. But you will very quickly find that if you have 100 of these, you can spend forever looking at this and you still wouldn't find it. So the key is to actually guide the pathologist or the researcher to particular regions of interest and to have him build something that is more useful than a 2D image. So we can actually generate a three-dimensional model where Daniel can explain this better.
Well, so, this isn't actually a realistic model. This is what we're doing here is we're taking some pretty basic techniques of, that we all know well, displacement mapping or light mapping or, even some good lighting techniques, and taking some of those layers of information that are just black and white maps and converting them to something else. And so rather than just visualizing everything as color, we can visualize some of these things as light. We can make them blink. We can make some of those things stick out. And then immediately, that picture that was just a sea of information of little colored dots becomes something that you can gravitate to. So all of those flashing lights are where there is growth, the cancer growth. And it turns out that they're in these kind of hardened shells that are surrounding the cells, and those are the things that we made grow. And so you can see the light inside of the walls lighting up, and this isn't realistic at all, but it is really easy for us to navigate because it's a 3D space, and we're really good at that as humans.
If you have biological questions, Jenny is here. She's from OHSU. She's actually a biologist and can explain this.
I don't know what she's talking about.
In order to revolutionize this, though, you need more than just visualizing, right? So we actually need to automatically identify each individual cell in that tissue, and then we can measure on those particular pixel locations what the expressions are of the different biomarkers.
Ideally.
And there's many, many millions of cells, so you cannot possibly do this interactively by hand. So we need machine vision to do that, and machine vision from facial recognitions and other applications. It's very good at doing certain things already because we have many decades into machine vision now that is actually helping to find the kites in this image and the people in this image. But there's also some dramatic examples on why this might fail. So Tesla car had trouble distinguishing this truck from a little white cloud in the sky. So you can tell that the machine vision is really not that reliable on its own, right? What we do is kind of look back and take an example here in nature. This is a mantis shrimp. It has the most complex vision system in nature that we know of. So you have two eyes, and they move together, and you can have three different sensors for red, green, and blue, and one for black and white. This thing has two eyes that move independent of each other. Each eye has trioptic vision instead of stereo vision, and each of those eyes actually have 16 different sensors. So it can see different colors of infrared light, different colors of UV lights. There's 4 different sensors for UV light. It can actually see circular polarization by quickly moving its eyes and actually do a polarization analysis. So it's going to get 4 Stokes angles and actually reconstruct images way beyond anything that you can do. read up on true facts of the mantis shrimp on YouTube, and you will know everything about it. It says this thing can see 9 more colors than you can. So anyway, there's a serious lesson in this, because if you look at the Tesla, why did that fail, right? Because it was relying on the vision sensors, and the vision wasn't good enough to distinguish that. So I don't know if Elon is listening, but the vision sensor is not good enough to actually drive a car on or to do pathology with. So, and this is coming from somebody who's doing image processing for 30 years. So instead, what you see on these self-driving cars, for example, is a plethora of sensors, right? There is a radar, a LiDAR, the thing on top is rotating. It's a light sensing distance measuring tool. There's things on the back, there are things on the front, and it takes in all these different signals, and that is actually how it is able to not crash into anything else. So we need to do the exact same thing by utilizing all these different biomarkers, these 100 different color channels. We can actually do much more reliable scene segmentation, much like the car is using 16 different input channels. We're using 100 different input channels to do better segmentation. And then once you know where the cell is, then you are golden, because then you can measure all these things, then you can make graphs of the data points, and now the researcher can actually point at regions in the feature space and highlight cells in the image space. Or you can highlight things in the image space, outline a tumor, and say, what's different about this in the feature space? And that's how you can learn what is happening. You can take that a step further, because you don't have to take the direct features, but you can use machine learning to actually find patterns and clusters, and you can make heat maps. You can do t-SNE analysis, you can do simple things like principal component analysis. All those things are available, and all those things will build statistical graphs or visualizations. And all the time you want to relate what is in the tumor region versus what's not in the tumor region, or what has changed on this patient over the course of time of treatment. And so we're looking at a piece of tissue, we're colorizing the nuclei of the cell, and the user is just basically playing with the gating on the various plots.
So computers are, we've been able to use just these kinds of tools to get the computer to see a bit better and distinguish these cells. And so we did the kind of work that you just saw. But computers aren't really good at seeing simple things like dotted lines, for instance. And so they have a hard time with that kind of thing. And this is what an electron microscope sees. Right? And it's all dotted lines. It's all dots. This is a really high-resolution image, but with really, with very few, let's say, raycasts. This is with, obviously, electrons for electron microscope, and there's not a lot of them casting. And so you get a lot of black space where there's nothing, no information.
Yeah,
and
so
computers aren't great at seeing that.
You go into pushing the resolution limits, the more you lose in signal to noise. So at this level, you're going to lose signal, you're going to get a lot of noise, and now it becomes more challenging to do the scene segmentation.
And what's amazing is a 5-year-old can see that stuff just fine, right? They have no problem discerning those lines. So it's time to teach these computers some new tricks. This is Maddie, my dog from a long time ago. She liked watermelon. So we've been teaching computers some new tricks. We've taught them how to play chess and how to play Go. And Google is teaching computers to understand doodles and recognize what these things are by asking people what those things are. And so actually, yeah, and so, there's lots of people interested in cancer. It wouldn't be cool if all of those people could help with this particular problem. And this particular problem, this is the current state of technology. Why is that not playing? Make it play.
Make it play.
Make it rain. Go. Why is it not playing?
So the current problem at Oregon Health Science University, where these images are being acquired, is that in order to outline and identify individual cells in these hugely complex electron microscope images, you have to actually... Go back.
It doesn't go back. Evil.
You actually have to, as a researcher, outline individual cells.
There you go.
And so this is a very tedious process because if you
Damn
can
it.
imagine a three-dimensional stack of these things with 600 images, you have to outline each cell on each of these individual images. And so that's where we would like to get your help. This is possible to do via crowdsourcing applications. So, and
Okay. Does this play?
Yep.
Will it play? It's not playing. And the next one isn't playing either.
the
That's
next
not
one is
helping.
playing either.
No, didn't play either. Okay, well, that's not good. But, oh well. We'll play it later because we're going to run out of time. There we go. So,
And this is
that's right.
nanoquill.org. It's playing right there. And this is my laptop, so it has no excuse whatsoever.
None.
So NanoQuill is a project that we started to help crowdsource this effort. So instead of having all the researchers color these images in, we'd like you to color these images in. And there is a coloring book that's actually a printed version and a coloring app that is an interactive app where you can take electron microscopy images and yourself color in the cells and the tissues. With a complete freedom, we actually let you color anything in any color, and then we get the images back for our research. So we get these colored results back that are then reassembled and optimized for our research, and then they are used to train artificial intelligence solutions to do this in the future.
Oh, let's go back for a
automatically.
second. The difference being, if you color it on paper, what you're getting is the actual images back, so quantity of images to train the computer. And it doesn't really matter what color is what, as long as they've defined outlines. If you get it back on the computer, we have the added bonus of knowing how people tend to see that drawing. So people will, a kid can see the continuation of that drawing and will draw it a certain way. And so we can capture all of those strokes and know how they're drawing it. So that actually informs the computer on how to recognize those shapes by being, moving continuously as opposed to just the actual pixels and shapes themselves.
So the images are printed or downloaded. They get colored by anybody. Then we get them back either via social media or via the upload on the nanoquill.org website. And so we get then multiple images colored by different people, one from Germany, one from Finland, one from France, and another one from Portugal. And then we can collect that information, reconstruct it on our processing side, and find the individual organelles. Even though we didn't tell them to find mitochondria, we can actually detect the mitochondria and use their human vision system to help out our computer vision system. And so that's one thing. So we made an app because the coloring book is nice for certain people so they can relax and draw with a pencil. The app is a little more fun because it's an interactive process and you can have some special effects. But then there's also the competitive aspect because we want people to keep engaged in this thing. So Maligna is a game that is going to be a competitive version of this where you can actually select cells for malignancy and swipe left, right. It's sort of Tinder for cells. And so what is next? The pathology workstation of the future is the key thing for us that's next. So what does that look like? This is, for example, a movie that doesn't play. Because it's probably the exact same, because it's a copy, it's not the...
Right.
So what happens here is that we color 4 different biomarkers as sort of the skin of the tissue. And then there are 2 types of immune cells here, one in purple and one in magenta. And they bounce up and down for the pathologist to see where the immune cells are in this particular tissue. So if you have a tumor and you have a certain treatment, immune cells are recruited to help cure that particular site. So it's very important to know whether they are recruited and how close they are to a tumor nest, and whether they build a wall around it and actually do their thing, or whether they are rejected because something else is happening that is rejecting the treatment from actually working at that location. And then we take that biopsy. This is a breast cancer patient who had metastasized in the liver. So the top sample is a liver biopsy taken before treatment, and the bottom section is a biopsy taken 4 months after treatment with a particular drug. And if you do so, then you can actually see that the CD8 cells have almost all disappeared, and the proliferation of the cancer has diminished a lot. And we can turn that movie on while we do questions so you can actually see this. So the other things are we would like to be able to do sequencing within a day and actually accompany that with that architectural and structural information about that tumor so that we can combine the genetic information with the architectural and the biomarker information. And ideally, you'll be able to do that while a patient is still in the operating room. So we want to have this in real time happening during surgery. So the time of this is critical as well. And with that, the visualization of that data needs to be fast and accurate. And that's where we need your help, as well as crowdsourcing. So come help us. We actually have a wall, a coloring wall at the QT and KDAP booth, number 1333. They were kind enough to share the back wall. So we actually have a NanoQuill image up on the back wall of that booth. And there are crayons that you can use to then color that in. You can take a picture and you can post it on social media. We'll retrieve it, and then it will go to OHSU for the research. And of course, you can find us on the emails below. And I will switch presentation while you start answering some questions.
Sure. Anybody have any questions? Yeah.
So, ever since you mentioned the computer, gaming has exploded and transitioned from just board games and races to virtual reality playgrounds. And it's been heavily reliant on that evolution you mentioned of CG and hardware. So do you see gaming field and other fields that are in similar positions sort of being anchored to that evolution, being channeled to answering questions in medicine, and answering the tough problems like that? Maybe you could expand a little bit on NanoQuill too.
Well, I think, I mean, there's definitely a connection for gaming in this. I mean, there's no doubt that there are a bunch of people that are very engaged in gaming. And just that engagement tends to be enough for us to get help, because they may not even be aware that they're helping when they're doing whatever it is they're doing in a game. So they don't even have to be interested in biology for them to be helping. So that's pretty amazing. I'm not really sure that it has all that much to do with technology in that sense, because the technology that we really need is just the back end kind of gathering the information. They could be playing Pong if that was helpful, right? Hey, that's Milo, my son. And this presentation took a long time and then took a short time to practice. Anybody else? Somebody else? Yeah.
I was going to mention kind of an answer to his question. There's a game called Fold It. I don't know if you guys
Could you talk to Mike? That would be great.
Yeah. So Fold It is a protein folding game. Adrian Truly, who was at CM at the time, put it together. Has anyone played it?
University of Washington, I think, was one of the.
Yeah, Adrian's back and forth between the two, but it's an amazing crowdsourced, and there's figures on 1 billion years a human would have taken to do it, and it was crowdsourced and solved some major protein folding issues in the medical community in
Yeah, I just want
months.
to bring that.
Yeah.
That was exactly right. I just want to bring that
Yeah.
up as an example, sort of ancillary to the problem of cancer, where they're crowdsourcing using game mechanics to motivate people intrinsically to participate in this research.
Well, and as Daniel mentioned, like it could be Pong. Like the people doing it knew that they were doing this,
The game mechanic
but the game was
much
fun,
less
right?
important as long as
they did it because it was fun.
Exactly. Yeah, and they built in a social component. I also wanted to mention a game called Remission, which to a lesser extent was. It didn't have to do with massive data sets, but it was a game that people could play while they were undergoing chemotherapy, and they found that there was a direct correlation to outcome, the quality of the chemotherapy's action against the cancer, as well as their adherence to drugs, was directly correlated to them playing this game where they're playing a nanobot fighting cancer in their own body. So there were some weird connections discovered there that I can see echoes of coming through here, particularly with deep machine learning and crowdsourcing this kind of stuff, which I think is brilliant, by the way. I also wanted to go back to sort of early on when you were talking about Illustrator. I'm a biomedical animator, actually, so this is right in my wheelhouse. I loved all of this.
Amazing.
There's still a need, obviously, to tell stories. We've learned already this week, we've heard a lot about story.
Sure.
And so this data set, translating this data set to an audience-appropriate methodology or communication pipeline, are these tools going to be made available to the general? I own an animation studio that does this kind of stuff, mostly for pharma, occasionally nonprofits, government institutions, things like that. This is the kind of resources that we're desperately in need of, because while we're telling stories using Hollywood-level CG graphics, we still need to have this touchstone of authenticity and accuracy. Are there ways that we can access this kind of stuff now, or is there a plan for that in the future?
That's why we're here, to find you.
Okay.
So if you keep your eyes peeled on our social media channels, SIGGRAPH Conferences on Facebook, SIGGRAPH Conferences and/or SIGGRAPH2018 on Twitter, we're going to include all of you in a conversation forever, for the next year, for the next few weeks. We will include examples of what's happening there. There's really interesting things happening with VR and medical recovery, with simulation, with nerve research, and we can share those with the group. And I'm not just going to tell you, you're going to tell me, right? We're going to post that and share it.
Fantastic.
So go and visit booth 1333.
Yeah.
Were you going to share?
Yeah, we will hang out there.
Yeah, we have coloring books.
Come
at the
color.
booth.
You have coloring books.
You can go get your own coloring book at
And
the
you
booth
can-
and save people's lives. So as I mentioned, for SIGGRAPH next year,
Thank you.
one of the first groups I'm talking to is someone that does exactly what he talked about. How do scientists tell their story a little better? A.J. Christensen is heading up that effort for us right now. If anyone believes that the world is flat, he's decided scientists aren't doing a good enough job of communicating. So, and anyone's ever seen Dancer PhD, there's some really great things out there right now. And I'm looking for all kinds of ideas to expand this program over the next year. Although both of our guests come from a lot of affiliations, they're both currently right now doing this work with a company called Quantitative Imaging. So when you saw their email, it was QI. So you can contact them directly or through us. Thank you so much for coming. Help yourself to beverages on the way out. We've got to turn this room over for whoever's next. Feel free to flood the stage and swamp these gentlemen as we turn over the room. Thank you very much. We'll see you tomorrow morning, 8 a.m. for Tony DeRose.